Before we begin, my name is Steven.
My name is Elena.
My name is Kaya.
My name is Angel.
My name is Diana.
Hello, my name is Preston.
Hello, my name is Mikhail.
Hello, welcome to our podcast, where we will be discussing our experimental design, which was phototropism and trophy rape seeds, which will measure its growth using a protractor. Phototropism is a way plants grow in response to light, allowing them to move towards the best light source for photosynthesis. It's how a plant finds the light it needs to survive. This process is mainly controlled by a plant hormone called auxin, which helps cells grow longer. When light hits one side of the plant, auxin gathers on the shaded side, causing those cells to stretch more than the ones in the light. That uneven growth makes the plant bend towards the light. The angle of the light can also affect how much the plant bends. When light comes in at different angles, it changes how auxin spreads through the stem. Steeper light angles like 39 degrees or 55 degrees can make the hormones distribute unevenly, causing the stem to curve more noticeably. By measuring how plants grow under different light angles, we can see how strongly light direction influences their movement. Auxin is made mostly in apical meristem, which is the tip of the plant shoot where new growth happens. From there, it travels down the stem through transport proteins, helping cells in the stem loosen their walls so they can elongate and bend.
My name is Mikhail. This experiment connects to the Radiolab podcast, Smarty Plants, and From Tree to Shining Tree. Both episodes explore how plants sense and respond to their surroundings, just like those stories describe plants communicating and reacting to signals. This experiment looks at how plants can read the direction of light and physically respond to it.
The aim of this investigation was to determine how varying angles such as 0 degrees, 39 degrees, and 55 degrees can influence the direction and extent of phototrophic growth in plants.
For our prediction, we think the plants that get light from the side will bend more than the ones that have light shining straight above them. When the lights come from an angle, the shaded side of the plant collects more of a hormone called auxin. That hormone makes the cell on that side grow faster, which causes the plant to bend toward the light source. So basically, the greater the line angle, the stronger we expect the bending to be.
The equipment consists of trophy rape seeds, 3 black solid trays, sealing tray, plug tray that contains 72 sections, Miracle Grow potting mixed soil, a protractor, a glass beaker that can hold 1 liter of water, a computer, labels with the numbers 0, 39, and 55 written on them, indicating degrees, measuring cup, tape, paper towels, and gloves.
Method. Step 1. Gather all materials listed in the equipment list, including the black plastic planting, seeding plug tray, soil, light source, and labels. Step 2. Label each tray according to its assigned light angle treatment, 0 degrees, 39 degrees, and 55 degrees. Step 3. Ensure the workspace has a consistent room temperature of 20 degrees and stable lighting conditions. Step 4. Fill each section of the 72-cell seedling tray with evenly Miracle-Gro soil. Step 5. Using a measuring cup to ensure each section receives the same amount of soil. Step 6. Plant 1 trophy rape seed in each section at an equal depth of 1 centimeter below the surface. Step 7. Lightly press the soil to secure it with each seed. Step 8. Measure 1 liter of water using a beaker. Step 9. Evenly distribute the water among all trays, ensuring each section receives the same amount. Step 10. Continue watering daily using the same volume and time to maintain consistent moisture. Step 11. Place each tray under the same lamp type and light intensity to ensure equal exposure. Step 12. Adjust the angle of the tray as follows. Step 13. Tray 1 should be directly above For tray 1, the lamp should be directly above the plants for 0 degrees. For tray 2, the lamp should be positioned at an angle of 39 degrees from the horizontal. For tray 3, the lamp should be positioned at an angle of 55 degrees from the horizontal. And for step 16, use a protractor and tape to secure each lamp at the correct angle and maintain it for the entire experiment. For step 17, make sure the trays are equal in distance from the light source to keep intensity consistent. For step 18, allow the plants to grow for a set duration of 7 to 10 days, ensuring all trays receive identical environmental conditions.
Our independent variable is the angle of light. We tested 0 degrees, 39 degrees, and 55 degrees. The dependent variable is a change in the bending angle, which we measured using a protractor. We found that by subtracting the bending angle from 90, we could figure out how much each plant curved toward the light. This helped us see how the direction of light affects the plant's growth pattern.
The controlled variables in our experiment were light intensity. We controlled it by using the same lamp for all treatments and setting it to one brightness level, making sure not to change that setting throughout the whole experiment. The reason for controlling this variable is because different intensities could affect the speed and amount of bending, making it unclear if the angle or intensity caused the change. Another variable was the amount of soil, in which case we put 429 millimeters or 0.43 liters. The way we controlled it was by making sure to measure each amount of soil and adding the same amount of soil to each seedling section. The reason for that is that unequal soil amounts could affect root support and water availability, changing growth rates within the plants. Another one was the soil itself, and basically the type of soil, in which you use the Miracle-Gro potting mix. We controlled this by making sure not to switch it with any other sort of soil and keeping that soil consistent throughout all the trays. The reason for this was that different soil types hold water and nutrients differently, which can affect plant growth. And then we also have the amount of water. We used 1 liter and made sure to water all the plants at the same time each day using a beaker or measuring cup, because more or less water can influence growth rate and health, affecting bending results. Next is the type of seed, in which we use the trophy rape seeds. We made sure to use the same species and plant seeds on the same day to ensure similar growth stages, because different seedlings may respond differently to light, affecting the reliability of results. And then we also have the container size and its shape. We controlled this by making sure to use the same exact seedling trays for all of the groups, because different container sizes can affect root space and soil moisture, as well as the growth patterns of the plant. And there is also the measurement method that we use, which was a protractor. We controlled this by making sure to use the same tool and the same measuring technique for each plant to determine the angle of bending. We measured the plant, and then whatever the measurement was, we subtracted by 90, because inconsistent measurements can make the data inaccurate or seem more biased. And then another one was temperature, and the way that we controlled this was by conducting the experiment in the same indoor location where the temperature was stable, in which case was in the biology room, where the temperature stayed at a consistent 22 degrees Celsius. And the reason for this is because temperature can also have an effect on the enzyme activity and plant growth rate, making the results inconsistent as well.
Here is the degrees of freedom, S statistic, and p-value. The data was analyzed using an ANOVA test to compare how different light angles, 0, 39, and 55 degrees, affected the degree of phototropic bending in trophy rapeseed seedlings. The ANOVA results showed a significant difference between groups, as shown right here, indicating that light angles had a strong effect on bending. Plants under angle light 39 and 55 degrees bent significantly more than plants grown under direct overhead light, which is 0 degrees. This supports the prediction that light coming from the side causes stronger phototropic responses.